Buenos días. Invitamos a toda la gente que está afuera que pase a tomar asiento, por favor.
Sí, Mauro. Si yo lo metí aquí, en ese punto como si yo no, porque sé que se menciona como ya no se van a escuchar correspondencias. Sí, ya no.
Buenos días.
¿Pero tu micrófono quién lo tiene?
Antes que nada, les pedimos una disculpa por el retraso. Algunas ahí. Les damos una más cordial bienvenida nuevamente al Coloquio Estudiantil del Instituto de Ecología, Conservación y Diversidad del Paisaje, Estructura, Conectividad y Resiliencia. Los invitamos nuevamente a apagar sus celulares para impedir interrupciones a los ponentes y para liberar la red. Pues hay una transmisión directa por internet y necesitamos la red libre, por favor. También le recordamos a las personas que nos siguen por internet, por el canal de YouTube, a que participen, mandando sus preguntas al correo coloquio.estudiantil.gmail.com o dejando comentarios en la página de YouTube. Para comenzar, contamos nuevamente con la presencia de la doctora Kimberly With de la Universidad de Kansas, con la charla A Top-Down Approach to the Experimental Design of Fragmentation Study, using natural landscape models to study habitat area and fragmentation effects on ecological responses. Welcome, Dr. With. Good morning. We welcome you to this second day of activity of our student colloquium. We also Sorry. We want you to turn off your cell phones to provide an introduction for the wireless time. To begin this morning, we have Dr. With second talk, so please go ahead. Thank you.
Buenos dias. Thank you so much for coming out for the second day of the colloquium. If you attended yesterday, my first presentation explored how we could use this class of models, these neutral landscape models, to define landscape connectivity, to quantify these critical thresholds at which landscapes become disconnected, and further how these can be used in a more theoretical context to explore species interactions with landscape pattern. And all of that was being presented in the context of evaluating the risk of invasive spread. So today, I'm going to do something very different, but still couched in the context of applications of these neutral landscape models. And just to provide some context for the type of research that I'm talking about, I think many of us are concerned about the relative effects of habitat loss and fragmentation. And most of this research, excuse me, this thing here. Very good, thank you. All right, we're back on schedule here. Again, most of the research that's been conducted on the ecological consequences of habitat loss and fragmentation, such as tropical rainforests, have been couched in the theory of island biogeography. And so of concern then has been the extent to which the differential effects of patch size and patch isolation influence species diversity, species richness within these habitat remnants. And so again, most of the research has explored these area and isolation effects on species richness. This is also, the theory of island biogeography has informed the design, not only field studies, but much of the experimental work that has been done to test these relative effects on species diversity. And so very commonly we will see studies that alter the size of patches as well as the relative isolation of patches in an effort to determine how these different elements of habitat fragmentation, habitat loss contribute to species, loss of species diversity. The idea here, I refer to this as a bottom-up approach to designing landscapes that vary in the presumed effect of fragmentation. And so by adjusting these patch properties, the hope is that we can create landscapes then that can serve as models of the larger landscapes that are being undergoing habitat loss and fragmentation. From an experimental design standpoint, however, there are only so many configurations that we can possibly do in whatever size area we have in which to work. In other words, we're limited in terms of the size of the area, the study area, in terms of how many different configurations of patch size, patch number, and relative isolation that we can feasibly explore. And so that potentially could limit the inference, inference space in terms of how we can apply this type of research, this experimental research, to understanding what might be going on in the broader landscape. So as an alternative, and let me just reference a figure that a version of which was shown yesterday, based on some of Lenore Therig's work, in which she's emphasized the distinction between the effects on patch structure that are due to habitat loss as opposed to that which is due to habitat fragmentation. And the important point here is that some of the effects that we generally attribute to habitat fragmentation, particularly patch isolation effects, could be due solely, and perhaps generally are due solely to habitat loss. That is, just a reduction in the amount of habitat can reduce patch size as well as increase isolation among those patches. And so we're left with the challenge of trying to isolate the effects of habitat loss from that which are due to fragmentation per se. And one of the consequences of habitat fragmentation is the creation of more patches, a greater number of patches, but depending upon the pattern of habitat loss, we could see potentially either an increase or a decrease in patch isolation, that is the distance among patches. So again, I just want to reinforce that one of the conceptual constraints, I think, that underlies many of these empirical investigations and experimental designs that ostensibly are looking at fragmentation effects are perhaps mostly looking at the effects of habitat loss. The other issue, and moving away from this sort of patch-based approach to studying fragmentation, is the recognition that fragmentation is clearly something that is happening at a broad, what we traditionally view as the landscape scale. That it's happening at broad scales. And so, given that this is a landscape-wide phenomenon, it makes sense that we perhaps study it at that broader scale. And so one approach to this that I'm going to talk about today involves the application of these digital landscape models as a way of developing a top-down approach to designing fragmentation experiments. And so rather than adjusting the properties of the patches in the hopes of devising these landscapes that vary in amount and degree of fragmentation, we can instead adopt a top-down approach in which we adjust the total habitat area as well as the overall degree of fragmentation, which is in the context of these neutral landscape models, this fractal algorithm that generates these types of neutral landscape models is just a single parameter. So by adjusting two parameters, we can independently adjust the amount as well as the degree of clumping, the spatial contagion or fragmentation, to create landscapes that differ in their patch properties. So again, the distinction I'm making here is the traditional patch-based approach, which is a bottom-up approach in terms of devising landscapes that vary in terms of habitat and fragmentation, versus a top-down approach in which we're adjusting the properties of the landscape, but nonetheless, which then results in different patch properties. And so we can see along this gradient of fragmentation that we end up creating patches that are smaller. We have many more of these patches, therefore more edge habitat, and an overall greater degree of fragmentation, as opposed to a landscape in which we've controlled or allowed for greater contagion. And so we end up with patches that are larger, therefore fewer of them, and less edge habitat, and an overall less fragmented landscape. So the idea then is that we can take these theoretical models that we can generate on the computer, and for those of you that will be attending the landscape ecology workshop on Monday, we will explore the software that you can use to generate these sorts of patterns in case this is something you might want to apply in your own research. But the idea then is that we can take these computer-generated landscapes and then create these in the field and produce this experimental model landscape system, or which are sometimes referred to as micro landscapes. In the informal discussion session after the poster session yesterday afternoon, we had a very good discussion about the fact that landscape ecology can be applied across a wide range of scales, that a landscape is defined in terms of the scale at which the organism or process when it's studying operates. And so by scaling these landscapes down to a more tractable level, we can also then determine how these processes interact with spatial pattern to influence the composition and the diversity of, for example, arthropod communities that I'll be talking about today. But this is by no means a new or novel idea. I was very much influenced as a student by this paper that was published by John Weems and Bruce Milne back in 1969. I remember there. But this was published in Landscape Ecology. And this is one of the first papers that really challenged the notion of landscapes as necessarily being a broad spatial extent, that is sort of an anthropocentric view of landscapes, but instead suggested that we could scale landscapes and adopt, since they were studying beetles, a beetle's perspective. I'll just highlight a couple of points here, where they're advocating for this micro-landscape approach, that this might then help to accelerate the development of quantitative as well as conceptual framework and understanding the influence of spatial pattern on ecological process. And remember our discussion and some of the talks yesterday, that landscape ecology is defined by the effect of spatial pattern on ecological process. And so from that standpoint, we can define landscapes at any scale and therefore can apply these sorts of concepts and methodological approaches to a wide range of scales. And so here again, they're advocating for scaling these landscapes to the sorts of questions or phenomena being investigated. So as I said, I was a student of John Means, and this was a very influential paper as I was starting my PhD program. It influenced the work that I did for my dissertation, but it's something that also influenced my approach to research. I think throughout my career, and so not all of my research has been theoretical, although at the time that we were exploring these sorts of interactions, there wasn't empirical or conceptual basis, other than, again, these either biogeographic or patch-based theory approaches of manipulation theory. And yet that didn't fully capture the complexity of landscape patterns and how species might be interacting with those patterns. So again, the theory development, I feel, was an important contribution, but then taking this to the next step and exploring experimentally how these sorts of landscape patterns interact with various ecological processes was a important next step. And so about a decade or so ago, when I was at Bowling Green State University in Northwest Ohio, a colleague and I took over a 4-hectare area of a former agricultural land that the university owned, and we developed this experimental landscape system. And just to help orient you, because I know it's like one of those worst tests where people see different things, but this, again, is showing the overall extent of the study area. So this area is approximately 4 hectares in size. But each of these little squares is a research plot, a micro landscape. And the green is clover that we seeded in these particular fractal patterns. Again, based on those computer-generated fractal neutral landscape models, we basically generated those on the computer, printed them out, and then went into the field and created this experimental system in which we have 6 levels of habitat area in our micro landscapes, varying from 10 to 80%. And then at 2 extremes of fragmentation, so very fragmented habitat distribution or very clump distribution. And so, excuse me. So for example, this would be probably an 80% clumped landscape. This might be a 20% more fragmented distribution. But well, here's a comparison here to get an idea of how adopting this sort of landscape or top-down approach does enable us to create a wide range of landscape patterns that vary in their patch structure that encompasses a large range of conditions in terms of habitat area as well as degree of fragmentation. And so we established this in the field. The study actually ran for 3 years and took a great deal of effort, as you might expect, to help maintain the interior of these plots. It looks white because it's, the soil had been compacted and so it dried when this aerial photograph was taken. And so we applied herbicides to the cells that were matrix that weren't supposed to have any vegetation. And then in between the plots, this gray area is just recently plowed soil. And so periodically we had a farmer come in and plow between these to try and keep the plots relatively isolated from one another by not having these vegetation bridges between plots. Of course, there are many arthropods that fly among these, so these are not completely isolated micro-landscapes, but it does help to try to isolate the effects of habitat and fragmentation on the sorts of patterns we were hoping to document. So this is the experimental design, the experimental model landscape system. And one part of this research was looking at some fairly fundamental questions related to teasing apart the relative effects of habitat amount and fragmentation per se, the degree of clumping on arthropod richness, and then also looking at different trophic levels. So looking at predators versus and parasitoids versus herbivores, whether one group or another would be more or less sensitive to habitat area or fragmentation effects. And then this interest in being able to scale up from patches to landscapes. Given that the finest scale in this particular system is a individual 1 meter squared clover cell, we were curious in terms of to what extent we might be able to take information obtained at that finest scale and then, say, multiply it by the amount of habitat to predict the overall richness at the scale of the plot or the micro landscape. So, if we're going to be doing these patch-based studies in the field or experimentally, we're assuming that we can scale them up to say something about species responses to the overall landscape pattern. And that may or may not be the case. Well, here's another view of the experimental system, just to, again, show the range of landscape configurations. But the arthropod surveys were quite intensive, and for this particular part of the study, it entailed going to each and every one of these clover cells and doing a visual survey, which means, of course, that we couldn't identify many of these species to a specific species level. Sometimes we had to make do with a coarser resolution, perhaps at the genus family level. But we can get a general and a very comprehensive view of morpho species richness, that is, the different types of species that are present, and use that as our response to these different types of landscape configurations. And so I'm emphasizing the enormous amount of sampling effort because my poor colleague was the entomologist, and so he was the one that conducted all of these surveys. And it could take up to 8 hours to survey just a single plot that had 80% clover. That would be a full day's work to go to every one of these cells. And I don't know, I think he's now in a psychiatric hospital from the damage that was done. But, his sacrifice for science is nonetheless appreciated. So the first part of this research is going to be based upon these types of surveys. And, just to get to the punch very quickly, there's a lot of research that has been emerging that shows that, in general, the amount of habitat on a landscape has a much greater effect, certainly on species diversity, but in many, on many other sorts of ecological responses. And so, in spite of the great degree of effort that we undertook to create these fragmented patterns, it turns out that in our own experimental model system, that, and I should point out that it's a very surprisingly rich system, given that it was in kind of an agricultural context in the Midwestern United States. So we encountered as many as 129 morpho species, which I realize from the standpoint of those of you working in tropical systems may not seem like very much, but for an agriculturally dominated system in the upper Midwest, that was actually quite a surprise. But again, we see this overall effect of habitat area on species richness. And so looking at the average number of species in these different landscapes that vary in the amount of clover habitat, we see this typical linear response where if we have more clover habitat, we have many more species. So the standard species area relationship. And this is how it scales on I'm not showing the log response here, but using the traditional formula for the species area relationship, this scales with the factor of 0.33. So that's interesting, but you'll note that there was also a significant survey effect. As I said, we sampled these landscapes over 3 seasons, and usually at least twice, sometimes 3 times per season. And one of the interesting things that did emerge was that in the first survey, so early in the development of this experimental system, as well as early in the season, this was the first survey during the second year, we saw this crossover effect in which species richness is higher initially in these landscapes that are clumped. In this first survey, this occurs at 40% habitat level. In the third survey, which was, again, an earlier season survey in that second year, it occurs at 50%. And then it goes back where we're seeing a higher level of species richness beyond that point in the first survey occurring at 60%, and again 60% here. So that was giving us this interesting interaction between habitat area and fragmentation, this sort of a non-linear crossover effect that occurred, as I said, either early in the season or early at the start of this experimental system. And so the only, for this type of survey, the only fragmentation per se effect that we saw occurred at these intermediate scales of habitat amount. So that's just important, I think, to keep in mind, to note, because we'll come back to that in a different context in just a few minutes. The second question that we were interested in was to explore whether there was any difference in response by different trophic levels, whether herbivorous species. I'm just showing some very generic examples of the types of arthropods that we consider to be herbivorous, things like the potato bean poplar, aphids, grasshoppers, and whether they responded in a different way to landscape pattern than predators or parasitoids, which are higher up on the trophic chain. And there have been some previous research that suggested that predators and parasitoids ought to be more sensitive to fragmentation effects relative to herbivorous species. And so to look at the response of these two groups, these two trophic levels, we once again see standard sort of species area curve where for either group, for either taxonomic group, we have more species, where when we have more habitat on these micro landscapes. But the important thing here is to note that the scaling effect in terms of the slope of this relationship on log plot is different. And in particular, it's a steeper slope for predators and parasitoids, and much of that admittedly is occurring below 40% habitat. So you can see how species are gained much more quickly, much more rapidly for predators and parasites relative to the slope for herbivores. And so that was interesting because, again, it might mean then that whereas they might not be more sensitive to fragmentation, certainly it appears that predators and parasitoids might be more sensitive to habitat area relative to herbivores, in that if we're thinking about this in terms of habitat loss, species would be lost, predators would be lost at a much greater rate, at a much faster rate relative to the expected loss of herbivores for a given decline in habitat area. And just like with the overall degree of species richness in the system, we see the significant habitat area effect and a significant survey effect. So there were differences from one survey to the next. But again, I think the important point to take away from this is that predators and parasitoids these higher trophic levels do appear to be at least more sensitive to habitat area, but not necessarily habitat fragmentation or fragmentation per se. The third general question was the extent to which we might be able to scale up from the grain, the spatial grain of the study, which was these individual clover cells, which were 1 meter squared, and predict the amount of species richness we might find at the overall landscape scale. So one expectation could be that if you have a cell, clover, 1 meter squared area within a plot with 80% habitat, maybe you would find higher richness within that context, as opposed to a 1 meter squared cell clover in a landscape with only 10% habitat. So if there was a correlation between the number of species within the individual clover cell, that 1 meter squared spatial grain of the study, we would see this linear increase potentially in the number of species that we find in the overall micro landscape at the plot level. So that would be the hypothesis or the expectation if we were able to scale from cell richness to plot richness. What did we find? This, which doesn't suggest that there's any relationship, or it's a very noisy relationship. Basically, these clover cells are sampling the broader arthropod community at random. And so there's really no, in fact, if you look at the overall average among all plots, or plot scale richness over all plots, we see an average of about 19, close to 20 species. And then at the 1 meter squared, the individual clover cell scale, generally we have an average of about 4 species. So there's no relationship. We can't simply scale up from these individual cells to predict how many species we might find at that broader landscape scale. And again, that should give us some pause in terms of thinking about the extent to which we might be able then to extrapolate from patch-based to landscape-based studies. Okay, so just very quickly to reiterate the main findings from the first part of this research. Habitat area effects trump, have a greater effect than the fragmentation per se on these arthropod communities. And the only fragmentation effect that we actually saw occurred at these intermediate levels. That's where we have these nonlinear interactions or nonlinear crossovers. Predators and parasitoids, these higher trophic levels, are more sensitive to habitat area effects, not so much fragmentation per se. And we can't scale up from the spatial grain of the study or these experimental plots to say anything about the total richness of species that we would find at the landscape or plot scale. So we do need to know something more about the configuration of these landscapes to be able to predict the richness. Well, we believe that these fragmentation effects come about probably as a consequence of edge effects, negative edge effects, in terms of, in some cases, the habitat edge might serve to either attract or to accumulate species, particularly if we have this kind of random assembly where species are more likely to come in and access a patch from the edge. But it's also, of course, possible that these edge effects could have a negative influence on species richness in terms of affecting the degree of competition or predation or parasitism that might occur. So there are different ways in which edge effects could either positively or negatively influence, for example, species richness. But we believe that these overall effects somehow manifest into a landscape-wide fragmentation response. And so the second part of this project of my presentation is trying to determine these relative effects, these direct versus what I call indirect effects of fragmentation on arthropod diversity in this experimental system. And one way to do this is to look very at a finer resolution in terms of whether diversity is different between clover cells that are located inside, a larger patch context versus those that have an edge with the bare ground matrix. And so whether or not we're seeing greater richness within the core habitat, and whether these communities are different. Do we find different sorts of arthropod assemblages if we're talking about edge cells versus core habitat or interior cells. So again, just to the terminology that I'm adopting here is that these are direct effects. What I'm referring to as direct effects of fragmentation is a landscape-wide property. That is the overall degree of fragmentation or clumping in the landscape, what is referred to as fragmentation per se. And so once we control for the effective habitat area, is there still an effect of the arrangement of habitat, the overall effect of landscape configuration on species richness or diversity? The indirect effects of fragmentation pertain more to these edge effects. And so these are effects that are that occur primarily at a local scale. The local scale here being the 1 meter squared resolution of these landscapes of the individual clover cell. And so again, because we believe edge effects generally give rise to these overall fragmentation effects, it would be helpful to determine, to the extent to which that's true, first of all, and whether there are certain domains in terms of the amount of habitat or the scale at which we expect to see predominantly edge effects, as opposed to overall fragmentation effects, again, defined at that larger scale. And so once again, we're dealing with the same experimental model system, but the sampling protocol was different for this part of the study. In this case, we actively sampled arthropod. Basically, it's a modified vacuum that you put a big cone that fits almost the entire 1 meter squared area of a clover cell, and you just put that over the cell and suck up insects or suck up arthropods into a bag. And then the real work comes later in the lab, where it took us actually many years to sort through all of these samples. And again, to try to identify these different arthropods, species, or family, as the case be. So we modified our sampling. We didn't try to sample every single clover cell. I would still be working on this if that were the case. But what we did was we selected 10% of habitat cells, just using a random numbers table. And then we sampled all of the landscapes with 10 to 40% habitat. And there were 3 each, probably clumped and fragmented at each of those habitat levels. And that, again, was because we had seen some evidence of fragmentation occurring at this intermediate level around 40, or we had expected edge effects to be particularly important at lower habitat levels. But then we only sampled 1 group of clumps, either fragmented or clumped at these higher habitat levels. And so it still was a tremendous amount of sampling. So we ended up sampling 243 of these cells and amassed an enormous number of arthropods. And as I said, it took several years to get through all of that. Just a reminder that even though this is a top-down design in terms of how we're creating these experimental landscape patterns, they do result in different types of patches or have different patch properties. So where we have these more clumped habitat or clumped landscape patterns, we end up with fewer larger patches with less edge, relative to landscapes that are more fragmented. They have an overall more fragmented distribution of habitat. And we do find that those have more smaller patches with more edge. Just to give you an idea, based on our survey, our sampling design, most of the cells that we call interior, these are cells, clover cells that were completely surrounded, that shared an edge with another clover, with 4 other clover cells. And most of those come from clumped landscapes, clumped habitat distribution. Most of the cells that we classify as edge cells, so these are clover cells that have at least 1 edge, and in some cases all 4 edges that are adjacent to that bareground matrix. And most of those types of cells did come from these landscapes that had an overall fragmented habitat distribution. Just to provide some sort of a conceptual theoretical basis for this, when should fragmentation effects be important? And there have been some suggestion based on some of one of our favorite simulation work and Andren's work looking at literature, that we might expect to see the configuration of habitat being important, particularly below a threshold of about 20%. Whether it's at 20% or 30%, not really important for our purposes here, other than just to point out that the configuration, the details of how habitat is arranged, they have been suggested to be most important at these lower habitat amounts. And that beyond that point, it was really the amount of habitat that was having the greatest influence on whatever the ecological response was. And either the number of species or the extinction probability, lots of different types of responses might be expected to show thresholds in these domains. And so given the sorts of indirect effects of these sorts of edge effects, when might those be important? And we can analyze the amount of edge in these different landscapes. And typically we find this sort of hump-shaped curve where the number of edges increases or is greatest at these intermediate levels of habitat. And this is true, it's plotting all these different landscapes from these very clumped habitat distributions to very fragmented and even random. They all show that humpback curve. It's just when you plot them on the same scale, some of these appear to flatten out. But if you could rescale this, they all show this sort of response where the greatest amount of edge is found at intermediate habitat levels. So therefore, we might anticipate that these sorts of edge effects, these indirect effects of fragmentation, would be most important at intermediate levels of habitat amount. Well, let's take a look at what we found in our experimental system. This is looking at the numbers of species as a function of habitat area in clump versus fragmented micro-landscapes. And again, we see we've been able to duplicate very nicely the species-area relationship. This scaling coefficient for a log plot. And so, once again, we find an overall significant effect of habitat area, but not so much fragmentation on the number of species that we find within these plots. However, if we look at just these plots below 40% or below, this is where we've got a balanced design, where we sampled all of the plots, both in the clumped as well as the fragmented landscapes. And if we just restrict our analysis to this group of plots, then we do see a fragmentation, a significant fragmentation effect. It just meets the threshold for statistical significance. But if you take a look at the data, you can see that really the significant fragmentation effect, the effect of the overall configuration of habitat, whether it's clumped, whether it's fragmented, is really only having an effect at 40%. The error bars are overlapping at 20 and at 10%. So I would argue that the significant fragmentation effect really only applies to 40%, landscapes with 40% habitat. So that's a bit higher than the 20 to 30% threshold at which fragmentation effects were expected to be important. And if we look at the local scale richness, that is just looking at individual clover cells, these 1 meter squares that are found within either a clumped landscape or a fragmented landscape, we can see that the landscape context overall does not affect richness, diversity, or evenness. There's no difference in the overall effect for landscape configuration, except for density, where the number of individuals per 1 meter squared area is somewhat higher in these clumped landscapes than it is in the fragment. But overall, there's no landscape configuration effect on the local scale richness. Remember, one of the other interests here is being able to look specifically at these edge versus interior cells to see if there's anything different in terms of that effect, that local effect on richness. And in this case, if we look at this local, the individual cells, and 1 meter squared cells, we do see significant effects between those edge versus interior clover cells in terms of density, richness, not diversity, but we do see a difference in evenness with these edge cells having somewhat more even communities than those in the interior. And so we do see these sorts of indirect effects, perhaps of fragmentation, in terms of whether a cell is surrounded by clover or whether it has an edge. And so that's getting at these indirect effects of fragmentation potentially. And they do appear to be important, even in this system, where if we look at the relative abundance of different species, these are the species that were most commonly encountered, and therefore were contributing most to the differences between interior and edge cells. We see that, for many of these species, their greatest abundance is found in the interior, but there are some notable exceptions. These leafhoppers and a conid wasp, a parasite, have marginally greater abundances along the edge, which is interesting, I think, in terms of understanding how maybe these clover cells, these patches are colonized, and many parasitoids, tend to occur where their hosts are located. So if the host species is located along the edge, then it stands to reason that maybe the parasitoid will be found there as well. So there's a significant degree of difference in the arthropod communities that are found in these interior clover cells versus those that have an edge. And we can break it down individually to look at these differences in communities. And these species here are just to highlight those that are predominantly or only found in edge cells, and a group of species here that are only found in these interior clover cells. So even in this very simple experimental model system, we see great differences in the composition of these arthropod communities, depending upon the location of the clover cell, in terms of whether it's embedded in a larger clover patch or whether it's along the edge of that patch. So the main findings then in the second part of this is that landscape fragmentation, the overall configuration of habitat, doesn't seem to have a very important direct effect on arthropod richness, except at that one level, what I'm calling an intermediate level of habitat, which is at 40%. But there still is the opportunity for these indirect effects, these edge effects, because remember, most of the interior cells, most of these cells that were being classified as interior, came from clump landscapes. Most of the edge cells are found in fragmented landscapes. And so there are these sorts of differences in the arthropod community, both in terms of density and richness, that are found depending upon whether we're talking about an interior versus edge cell. And so we do see that there are these sorts of indirect effects. But that still doesn't quite answer the question of whether or not these edge effects might be driving some overall fragmentation effect, at that 40% level. And so just to kind of create a conceptual hypothesis here, remember that when we look at the amount of edge on a landscape, it is greatest at these intermediate areas of habitats. And see this pump-shaped curve. And what the results of our study suggests is that below about 30%, so below that 40% threshold that we found, we found very strong effects of edge. This is where, again, we have a lot of cells that have edges in these fragmented landscapes, particularly when there's a very low level of habitat. But there was no overall fragmentation effect for those 20% or 10% landscapes. We have those overlapping error bars in that domain. And so here in this domain, at these low levels of habitat, below about 30%, which is, remember, where theoretically fragmentation was supposed to be important, it instead appears that it's these patch properties that are most important. That is the size of the particular patch, the relative isolation of the patch on the landscape. But it's a patch property, not an overall landscape property. That's important, because remember, the overall landscape property of fragmentation configuration was not significant for these landscapes at 10 to 20% habitat. So I'm arguing that it's really these patch-based properties, the patch size, patch isolation, that is important in this domain. And this is more of an indirect effect of fragmentation. It's not a fragmentation per se effect. It's an indirect effect because, again, if we have very little habitat, we're probably going to have a lot more edge regardless of how it's arrayed on the landscape, whether it's clumped or overall fragmented. At the other extreme, and I've just arbitrarily here said it at 70%, we have very little in the way of edge effects because the habitat is sufficiently abundant. There's no overall fragmentation per se effect. Landscape configuration doesn't matter. There's so much habitat out there. You don't have a lot of edge. The habitat isn't really fragmented per se. And so in this domain, the overall effect of habitat area dominates. And so what we're left with is that it's in this intermediate range, certainly at 40%. I don't know, how wide this range might really be because we didn't have a balanced sampling design, at least in our study, to look at 50 and 60% clump versus fragment. But certainly at 40%, we saw that direct effect of fragmentation, that landscape configuration, whether we have clumped or fragmented landscape, had a significant effect on species richness. And so it's at this scale that fragmentation appears to be most important. And this then is a direct effect. Edge effects, can be mitigated by the arrangement of patches, whether we have an overall clump landscape or an overall fragmented landscape. And so this is sort of challenging, again, this idea, about where fragmentation ought to be most important. There's been a great deal of emphasis on distinguishing between habitat area and fragmentation effects, fragmentation per se effects. This suggests that we should also take some care in determining whether we're talking about fragmentation per se, which is a landscape-wide property, versus some of these edge effects, which are more a patch-based property and are perhaps an indirect consequence of fragmentation. But again, it's another layer of distinction, and which may then affect our ability to extrapolate from patches to predict the effects of fragmentation at a broader scale. Okay, with that, I will conclude, and thank you for your attention, and if we have time for this, I'd be happy to answer any questions.
Recordamos la dinámica de preguntas. Primero son algunas preguntas en el auditorio, después si hay preguntas externas y finalmente por el canal de YouTube. En el auditorio tenemos alguna pregunta. Again, thank you very much for the presentation. In your first experiment, you found that predators, parasitoids were more sensitive to habitat loss. In your second experiment, did you find a difference between herbivores and predator parasitoids between age and interior cells?
I'm afraid we didn't examine that particular level of response. It took such a long time just to get the overall community response that we did not explore that. So I think that would be a good future analysis to do. Yes, thank you.
Great talk. In some of our work, we found that fragmentation effects are more severe on less mobile organisms, more mobile organisms, integrated across heterogeneous mosaic. You look at mobility as a factor in this study.
Yeah, good question. Not for the overall community response. Again, part of the thinking here was that the system was being colonized naturally. It's basically passively sampling the surrounding agricultural species pool, arthropods. In another type of study that came out of this system, we did examine the ability of a predator, two predators, two ladybird beetles to aggregate in response to the abundance and distribution of their aphid prey in these landscape systems. And there we did find a difference in the sensitivity of these two predators based upon their ability to aggregate in response to the distribution of aphids as a direct result of their relative mobility. A native species of coccinella was a ladybird beetle, moved very rapidly from stem to stem, but didn't tend to fly very much. And then this introduced coccinella, the Asian ladybird beetle, which was introduced specifically for the purposes of biological control in these agricultural systems, would crawl up a clover cell and then fly to, to another clover cell, either right next to it or another patch in the same landscape, and even to other flies. And so we were able to document, at least for those two species, that mobility did play a big factor in terms of their ability to respond, maybe not so much to the landscape pattern as it was to the distribution of the aphids, but the aphids were responding very much to the landscape pattern because once they arrive, they no longer disperse. So it's more of an indirect landscape response. So only for that particular group of species.
We have a common question here. Let's see if I understand. What about in places like you have big areas where there is no natural surroundings, that everything is transformed into agriculture, different types of agriculture, and you have grasses and cities. So what about their habitat?
Well, this particular experimental system was done in a highly modified landscape. It was dominated by agriculture. We had different row crops, soybeans, corn, And we haven't looked at whether in a given season or a given survey, because this did vary from year to year, whether it mattered, whether there was corn or soybean. But this was a highly, transformed landscape. It had once been a big swampy area, swamp forest, but that had all been transformed 150 years earlier. So I'm not sure that in this case, we're still seeing, I think it's remarkable that we see any response to habitat area fragmentation, because the species that we're dealing with are, they're fairly generalists, I think, very characteristic of these agricultural systems. It's possible that for some of the small remaining woodlots in the surrounding landscape that we're getting an occasional species, that flies in, but it's mostly a very generic arthropod community that you would find in that sort of agricultural landscape context. So, things be more interesting if there was, again, a different landscape context. Yeah, I would think so, but I don't know how that would alter the results.
¿Tenemos preguntas externas? ¿Preguntas externas?
What happens in terms of conservation with the result that between 30 and 70% of Alta's area, the effect of fragmentation is very important. We should give some interpretation in terms of conservation.
Yeah, that's a great question in terms of what would be a implication of this finding if it holds. I mean, we have no way. I don't know whether this is a general finding, but one implication of that might be that we can perhaps best address these sorts of fragmentation effects in these intermediate levels of habitat loss. So our best opportunity in terms of managing landscapes to preserve intact habitat would have the greatest effect on species diversity within that domain, approximately 30 to 70%. I don't really know how wide that is, because as I said, we don't have the data, but somewhere presumably in that intermediate range is where we would expect, managing the landscapes at that broad scale would potentially have the greatest effect. Whereas at, 20, below 30%, you're really dealing probably with isolated fragments anyway. And so that might be an indirect consequence of habitat loss and possibly fragmentation, but perhaps that demands a different approach. It's not so much landscape management as it is, fragment management, patch-based management.
Gracias. Agradecemos a la doctora Witt por sus dos pláticas. Si a alguien le quedó alguna duda, pues va a seguir aquí con nosotros y de hecho el lunes hay un taller. Y bueno, a continuación, el doctor Samuel Cushman del Servicio Forestal de Estados Unidos nos presentará la ponencia titulada Landscape Genetics.
Okay, thank you, Dr. Reed, for all of your talks, very interesting. And hopefully, well, if anyone that would want to know more about the team, she will have a workshop on Monday. So anyone who is interested, come with me so we can register. And now we're going to have our next talk with Dr. Cushman, who is also going to talk about landscape genetics, and he will also have a workshop colloquium Tuesday. Okay. Thank you.
All right.
Welcome, Dr. Cushman.
Yes. Thank you very much. Glad to be back here today. The topic of this talk is landscape genetics, which is the fusion of landscape ecology, understanding patterns and processes and interactions across scale, habitat loss and fragmentation, merging that body of theory and research with population genetics, which is trying to understand the dynamics of populations and its effect on genetic diversity, genetic differentiation, and adaptive evolution. And of course, evolution happens in ecosystems, and ecosystems are spatially complicated at a variety of scales, temporally varying. So it would seem obvious that understanding population dynamics, population genetics, and evolution would require a landscape perspective. But perhaps to our surprise, that has not been the case until the last few years. And it's a simple matter of the great power of 20th century ecology was mathematical modeling. And the mathematical models that developed population genetics, which many of you probably know, are based on simplifying assumptions that allow you to develop simple algebraic equations to predict things like heterozygosity, genetic differentiation, selection, and equilibrium between gene frequencies with different levels of selection. But those things all assume homogeneous, well-mixed.
La base de datos de virus ha sido actualizada.
So what happens if you insert spatial and temporal complexity into this process? And that's what landscape genetics is all about. It's truly a revolutionary change in advancing our understanding of genetic processes at multiple levels. So one of the things that I do is I'm the specialty chief editor for the journal Frontiers in Genetics, Frontiers in Evolutionary and Population Genetics in particular. And I wrote a little thought piece last year about the grand challenges in evolutionary and population genetics. And from my perspective, the great challenge is integrating several things, which essentially are integrating landscape ecology ideas and pattern process relationships with genetic theory and genetic data. And in particular, there's a revolution in the world of genetics related to genomics and epigenetics and bioinformatics. In the very recent past, genetic information was very sparse, very expensive, and very uninformative because of the challenge of getting meaningful data out of DNA. But genomics has changed everything. We can now very cheaply and quickly get thousands and thousands of reads and markers out of genetic samples. And epigenetics revolution, that actually gene regulation, gene expression might have a much bigger effect on fitness and evolution than genetic coded variation itself. And the challenge of integrating all this bioinformatics, we have databases that are terabytes in size, just genetic data from a few individuals, integrating that and analyzing it in the context of its feasibleness. That's the challenge. But these are just numbers. Genomic data is just numbers. Epigenetic data is just numbers. Bioinformatics just stores and curates numbers, To understand that in an ecological sense, we need experimentation, the kind of things that Dr. With just showed us, experimental model systems, for example, where you control the patterns and then investigate their effects on processes. And modeling to generalize and extend those insights that you can gain on mechanisms of controlled experiments to broader scales of space and time. So the big challenges are integrated genomics, experimentation, model, intersection of these things. And biggest challenge is these are all fields that are done by different people, different traditions, often different departments, different terminologies, different methodologies. These people often don't know each other. They don't often work together. The biggest challenge in advancing ecology, genomics, landscape genetics is a transdisciplinary challenge, crossing these boundaries to have experts from all of those topics working together on a common field. So the basic idea is data without models are not informative. It's just numbers. Models are a process that explains the numbers and predicts the numbers. You need models to make data informative. But models without data are not compelling. We can make up models based on theories and hypotheses. They might be very interesting. You need data to verify. Experiments without models are not expensive. You know, in a controlled experiment, we can identify mechanisms. But those experiments are usually at a small scale, over a short period of time, controlling the few parameters explicitly, and they are not able to generalize. So models allow you to generalize by saying, if this relationship is true, and if we apply it to a broader scale of spaces, and we're modeling in our experiment, what would be the implication? In models without experiments are not verifiable. Experiments allow you to verify hypotheses you can generate from models. Models allow you to explore a huge range of possibilities of pattern process relationships. But experiments allow you to verify those things. So this is a great unity of this idea. We need the rich data and genomics. We need models that allow us to explore pattern process relationships theoretically. We need experiments to verify and confirm those relationships. So, first grand challenge I identified in the evolutionary population genetics is how to move toward stronger influences in population, landscape, conservation genetics. And in conservation genetics, we have this tradition. And it's not unique to conservation genetics, but might be more common in conservation genetics than maybe other sub-disciplines in ecology. And it was the sort of verification is idea of, I have a hypothesis that this road is a barrier to gene flow. And so what people did is they sampled genetic structure on one side of the road and on the other side of the road. And they said, there's a difference. Therefore, the road caused the difference. And that's not good inference. It's an idea called affirm to be consequent. It's a logical fallacy. And it goes like this. We have a theory A, to explain a phenomenon. Maybe it's this road caused genetic information. If that theory A is true, then fact B is the result. Fact B is there's differences in the genetic structure on either side of the road. We observe fact B, there are differences on either side of the road. Then we assume theory A is true. We assume that the road caused it. That's a fallacy. Why is it a fallacy? Well, think about our bear example from yesterday. Here. We had two known models, isolation by barriers and isolation by distance. And if we had done what everybody had done before our paper, This is the first paper that did this more comprehensive evaluation of many hypotheses in optimization. Every other paper had done this. With the theory, isolation by variables. If that is true, then there would be a significant relationship between genetic structure and our variable hypothesis. Well, it was a significant relationship. It was very highly significant. And we would probably have assumed the variable model was true. Very common in conservation theories. We have a second known model, isolation by distance. That's this. In conservation genetics, up until the last 10 years, almost all the papers were a barrier hypothesis or a distance hypothesis, and usually only testing one. So we had the distance hypothesis, and the distance model would be significantly related to genetic differentiation. It was significant. It was very highly related. Therefore, we assume isolation by distance. Very, very common in the conservation literature. But of course, you see, it cannot be both. They're completely logically incompatible. That an isolation by distance, where there's uniform decay in genetic relations with distance, and isolation by variance, which assumes hand-picked population in one mountain range, hand-picked population in another mountain range, and a variance. So this is something that makes us nervous when we see two completely contradictory hypotheses getting basically equal support if you're looking in this verification, this idea, just looking for a correlation consistent with your observation. So in our analysis, we tested 110 models. They were all statistically significant. Genetic structure was correlated with every model. But you might recall, we used this causal modeling idea. We competed all these models in a framework that we can rank their relatives. We found that the barrier model was very poor, relative. 102 out of 110. The Euclidean distance model was relatively more, 35 out of 110. With this causal modeling, we can reject the barrier and distance models. Confirm that a landscape model is most consistent with the observations, mineral elevation, forest cover. Then we can rank those landscape models. So this gives a big step forward in addressing ways to make stronger inferences in conservation and landscape genetics. It's really surprising when we look at this. It seems self-explanatory that you have to compete and compare a reasonable number of hypotheses. It's a very bad inference to just say, I think this is true, I'm going to look for evidence that's consistent with it. I find evidence that's consistent with it, and then I say that's true. That's very bad practice. But in the field of conservation, almost everybody was doing it for 15 years. It's not only in conservation, something to be aware of. So yesterday I showed you the variable competed 110 hypotheses and found one that's best. This paper by Andrew Shirk on mountain goats. So the next step. And what this paper did is use the same general framework of looking at a range of different values for a bunch of parameters, evaluating the support for each one. But in our case, we had 110 hypotheses as sort of a factorial cube. And Shirk actually went through and did an optimization, which allowed him to explore a much more broad hypothesis space. So, for example, he began each with an expert guess, an expert opinion about how each parameter would influence gene flow resistance in mountain areas. Distance to escape terrain, roads, land cover, elevation. And did a univariate optimization, changing each of these parameters in terms of the relative resistance and a functional form of the relationship. And found that in all cases the experts were wrong, and sometimes very wrong, but did univariate optimization. for each variable's relationship with genetic differentiation independent, which is good. But the response of a species to the environment is not a univariate response, it's a multivariate response. And in combination, the relationship between the landscape structure and gene flow could be different in a multivariate context than optimizing each variable separate. So even through several rounds of multivariate optimization, starting at the parameter values of the univariate optimization and then holding them all except one constant at that, and then varying that last one until it was optimized, and then going to the next parameter and varying it until it was optimized, and going through that iteratively until you reach a stable solution. And found the multivariate solution for most of these variables was different than the univariate solution. And that allowed us to develop a fairly sophisticated and much more detailed understanding of the amount of gene flow in relation to the landscape than the idea we had in the variable, which is a very simple factorial hypothesis. 110 seems like a lot of combinations. This analysis is thousands and thousands and thousands of combinations of parameter values, which allow you to be much more finely optimized. So the idea is this multivariate optimization can identify topographic complexity, water bodies, and aspect. And then we can do simulation modeling to identify the correct drivers. So what we were able to do, I think was a big step forward in how we can optimize the relationships between landscape structure and genetic differentiation. Now, of course, people are really, really like and want to use is this idea of multiple regression of distance matrices. And all of the data that we use in landscape genetics is these pairwise matrices of genetic differentiation and cost distances between observation points across each of our variables. And we were using this optimization approach using man-tele tests. There's some limitations to that. And people want to use regression. Regression is the workhorse of modern ecological statistics. And the generalized linear model framework is very powerful. It allows you to do a lot of things. For example, we can use this standardized statistical framework to correlate responses with genetic distance. You can use things like AIC to robustly do model selection based on parameter number. But there is a problem. When you add spurious variables to a regression, variables that are not related to your process, AIC values go up in a node-based AIC, not a distance-based. That's what almost all people use, comparing this point to other points in values around that point, predicting. So predicting species occurrence at sites based on the characteristics of that site, like most professional ecology users. This is what you want to have, because AIC penalizes models for spurious predictors. And the lower AIC is better, and it should go up when you add spurious predictors. The problem is, in distance-based AIC, it goes down even when you add spurious predictors because of the non-independence of the samples in a distance matrix. So, this means we are not able to use AIC, and we're not able to use generalized linear modeling in the same way that we do in main line code. So, it's an area we may need to be developed, and I do believe still now the best method for doing landscape genetic optimization for gene flows, Andrew Shirk's method of optimization that I showed you for now. With hope the regression framework of AIC will come into use when it is refined and improved, but right now it is not reliable. So our second grand challenge could be developing and applying advanced individually based spatially explicit genetics models, because these models, as I said, are critical, because data without models are not informative, you just have numbers. The models have great power in that in a model you can control a process. What I mean by that is when you're doing research in the field and you're collecting empirical data, you never know the processes they are in action. You infer them through induction by correlating what you see with something else, and you find a relationship, and then you can hypothesize that that is a process that's right. But you don't really know. The model allows you to control the process. You can say, I know dispersal is controlled by these factors, and the organisms can disperse this far. And therefore, what is the result if that process is true in different situations? And we developed a model called CPOP, which is spatially explicit cost distance population genetics model. We learned something about it on Tuesday in the workshop. My PhD here in Langdon, Belgium. And the idea is to predict genetic patterns and stipulate and control dispersal processes. So in this case, you control the process and you simulate the pattern. Where in the empirical work, you observe a pattern and you infer a process. This gives us great feedback between the empirical and the simulation. And so the CPOP model, spatially explicit individual-based population dynamics population model, well, length, uses resistant surfaces and ideally in inheritance, where he reproduces, disperses organisms on the landscape as a function of movement, reproduction on a continuous cost surface. So what it does is you can place out a hypothesized distribution of organisms, in this case, hypothetical bears in our place in northern Idaho at approximately the distribution of density of bears. There are a lot of bears. And you then create this cost distance matrix, as we had seen before, but now not between 146 real bears, but in this case between about 1,000 hypothetical bears. And then you can use this cost distance matrix to simulate it. Control how long we run it, what the movement function is, there's probability of moving to a place decline linearly, or is it inverse square, or some other function cost distance. So we're going to use this model to explore its implications and see what is different in changes in, for example, heterozygosity. When you change from a pan-metric model to Euclidean isolation by distance, barriers, and landscape resistance. Equation 1 is what you expect over time as heterozygosity declines, basically as a function of effective population size linearly. In the pan-metric population, our model works perfectly for that, but it declines more rapidly and as you get non-ideal populations isolated by resistance and barriers and landscape structure. So this gives you an idea of how you can begin to explore theoretical implications of landscape energy in individual-based models. I'm going to show you a few more things now and how you can apply that. So our third grand challenge that was how we can evaluate the performance of different statistical methods to estimate and measure the effects of landscape features on genetic differentiation. And so, as I just described, When you do empirical work, you observe a genetic pattern and you infer the population process through an induction, statistical model, to associate. The simulation allows you to say, if that population process is true, what genetic pattern would result from it? And this interplay between them can allow you to evaluate how well a statistical method for induction to infer a population process actually works. Because often people don't really know. They just say there's a correlation, therefore it's true. It may not be true. So this paper allowed us to evaluate the performance of our method or our variable that we used in this causal model. And you might recall we had three main hypotheses in that analysis. We had isolation by distance with uniform resistance across the landscape. We had isolation by a barrier in this valley with pandemic populations on either side. And we had isolation by landscape resistance with lowest elevation and middle elevation forest, lowest resistance in middle elevation forest, high resistance in low elevations. And so, when we use our simulation model, when we stipulate and control the landscape resistance model to be the driver of genetic differentiation, we simulate movement of bears as a function of that landscape resistance model. Over time, the correlation between the cost distance and the genetic differentiation increases. It flattens out at a very high correlation, about 150 generations. That's what you expect. If that process is driving genetic structure, you should see that effect. But when that process is driving genetic structure, we also see a correlation with distance. We also see a correlation with barrier hypothesis. And these correlations are equal to, almost equal to, that with the true process. So this is the affirming the consequent idea. In the past, almost every study has asked an isolation by distance question or an isolation by barrier question. In our system, if the landscape model is true, people still could say, I see a strong correlation with distance and I assume distance or with barrier. So there's a huge risk of affirming the consequent in the distance and barrier hypothesis. These are spurious relationships. And a correlation that is consistent with the hypothesis does not demonstrate its causality. We know that. And there's very high probability of incorrect inference due to a naive correlation analysis. But you might recall, that's not what we had done in our variable. We had done this. Causal modeling of distance matrices. And we said provides a means to test which the landscape barrier distance model is correct. And when we used our simulation models to evaluate this, we found that yes, when the distance model is stipulated as the true process, isolation by distance is the only thing driving beam flow. There's no relationship with barriers and no relationship with landscape resistance. We could identify it as correctly driving and reject these two goals. If the barrier model was stipulated correctly as a process driving the gene flow, and we simulate the resulting genetic structure, we could identify it correctly and reject spurious relationships with distance and landscape. And the same way, when we stipulate landscape as the process controlling gene flow, the landscape resistance drives movement in dispersal. Then we can reject the distance and barrier models. So we're able to show that this causal model approach, even though there are very, very high spurious correlations with these alternative hypotheses, we could correctly identify the true drivers with very high power. But we had an additional question that was only competing with three fairly different things: isolation by distance, isolation by barriers, and a landscape resistance model. What we're really interested in is how we can select among different landscape resistance hypotheses. And maybe thousands of possibilities for how a landscape could influence heat flow. What statistical methods allow us to identify the correct one? And so, recently, last year, we wrote a paper reevaluating causal model and health tests. And our question was, does this causal model approach identify a wide range of resistance models as the correct drivers when they are controlled in a simulation model made to be the true process? And here's a figure which explains the problem we have. Here are 36 resistance hypotheses which combine forest cover, elevation, and roads, and their effect on resistance. And this is the correlation between them in terms of how similar the resistance values are across that landscape between these different hypotheses. And this color bar ranges from 0 in blue to 1 in red. And you see this entire surface is basically red in orange. Correlations of over 90% between the alternative hypotheses. When things are correlated over 90%, can you separate? Hard to do. They almost completely identical in the information they carry. And this is the problem in landscape genetics, because all the resistance hypotheses are usually highly correlated. So we're able to evaluate how frequently we make inferential errors based on how correlated hypotheses are. Two kinds of errors you make. Type 2 error, failing to see an effect when there is one. Type 1 error, spuriously seeing an effect when there isn't one. This is type 2 error, and this is correlation between alternative hypotheses. This is the probability of type 2 error. You see, you have basically no chance of being wrong with type 2 error. You have no chance of failing to see a relationship. Independent, the true hypothesis independent of an alternative hypothesis, until that correlation between those models is above 0.1. So it's very, very, very high power to see an independent effect of the true hypothesis compared to alternative hypotheses. Type 1 error is a much bigger problem. Type 1 error is seeing a spurious relationship with an alternative hypothesis, one that is not true, independent of the hypothesis that is true. And that happens even when there's low correlations between alternative models. And as we get to high correlations, there's a very large chance of incorrect inferences. Failure to reject spurious hypotheses. So we developed something we call reciprocal causal model, which allows us to compete all these hypotheses based on their relatives. And it basically allows us to say, I'm looking for the hypothesis that is supported independently of all other hypotheses, and no other hypothesis is supported independently of it. And given that I'm behind schedule, I'm not going to talk in more detail, but we can explore it further in the workshop. And another recent paper that we developed to compare different methods for inferring barriers to heat flow. A lot of different methods people use. Clustering methods, edge detection methods. And this paper was important because it showed that there's a big difference in the performance of different methods, and many of them are exceptionally vulnerable to type 1 errors like I was just describing. Which is a topic I'll be able to talk much more about if you're interested. But our fourth grade challenge was evaluating time lags in landscape genetics. Populations have inertia. Individuals often live for a long time, and it takes quite a long while for the genetic structure of the population to respond after the landscape has changed. So we are interested in exploring how different statistical methods performed and how sensitive they were to detecting changes in the landscape and how they are affected by climate change. So we were paper to explore it using a simulation model. And we found a very big difference between individual-based methods and population-based methods. Classical population genetics uses population-based statistics, assuming panmixia, using things like FST to look at differentiation between populations. Individual-based methods looks at individual differences in the genetic structure among two paired individuals, and is much more sensitive to recent landscape changes. So we found that individual-based methods are much more powerful and sensitive than population-based methods and respond much more quickly. That was an interesting observation. But they both have time lags. The time lags are an important matter in landscape genetics. If you are interested in detecting the effect of a recent barrier, it might be necessary to look at movement data itself rather than genetic data, or to look at parent-offspring pairs, or using genetics to identify displosive events rather than the genetic structure of populations because of the climate. Another grand challenge in landscape genetics is evaluating alternative sampling designs to see how they affect our ability to detect the relationship between landscape structure and population processes of genetic differentiation. So we did a simulation of this, looking at how the number of individuals sampled, number of genetic markers sampled for individuals, and the allelic richness of the genetic data set, how many alleles per marker there are, on our ability to detect spatial genetic patterns. And we found that when there are few genetic markers, each of them have few alleles, it's very hard to detect genetic effects. And so what we would like is to develop research projects where we have, in advance, designed them so they can succeed. And that usually involves doing a lot of markers with a lot of individuals, particularly when there's low genetic diversity in the population, as in many rare species. So if there's rare species with low genetic diversity, lots and lots of genetic markers can be a big improvement in our ability to detect things. Another grand challenge was evaluating the effects of limiting factors on genetic differentiation from types of landscapes. Limiting factors are a big deal in philosophy theory. And it works like this. In Idaho, where we study our bears, this is the valley, it's thought to be a barrier. These are the mountains, middle elevation forests, good for bears, movements, the highest elevations, and the lowest elevations of the mountain. So we detected the elevation relationship, and we detected the forest relationship in this landscape. And then our good friends, not very far away, studied bears in this landscape. What do you think they have? Nothing. Because this is all middle elevation. It is all forest. Elevation doesn't limit variability in that landscape. Forest cover doesn't limit variability in that landscape. It acts like isolation by distance. But that does not mean that bears don't need forest cover in the elevations. It just means you cannot detect the relationship in a landscape where that's not limited. So we explore that further in a paper here, where Ruth Shortbull repeated our bear study, she had done in Idaho. She repeated 12 different landscapes, which varied with extent, forest, the variation in elevation, and the number and density of roads. And we found, not to our surprise, you only detect the effect of forest cover when it is limited, when there's a relatively modest amount of forest and when it's highly broken. You only detect the effect of elevation when there's a lot of variation. If it's a flat landscape, you detect no elevation effect. You only detect roads when there's quite a lot of roads. So when these things are limited, not a surprise. But we wanted to say, okay, well, what can we do with that? Can we generalize that? Say, when will things become limited? So we use mutual models. The same ones that Dr. Wynn was describing, varying habitat extent, habitat fragmentation, and apply landscape genetic simulations to those. And then we're trying to understand the relationship between the extent of fragmentation and habitat and our ability to detect the effect of the resistance of non-habitat. Assuming that habitat is low resistance, when you detect the effect of non-habitat as being resistant to movement as a function of how much non-habitat there is, how fragmented. And essentially what we found, I'm only going to show you one of these, because these are different levels of population size. But essentially, we found that the probability of detecting the landscape genetic effect that is there, because we control it in our model, we know it is there, that non-habitat is resistant to movement, is very, very, very low. You have low patch density, and you have high correlation length, which is connectivity of habitat. So the habitat is not fragmented, low patch density. Your habitat is very extensive and connected, high correlation length. You cannot detect a landscape genetic effect because non-habitat does not limit gene flow sufficiently to structure genetic differentiation. When you have high fragmentation and low connectivity of habitat, you have a high chance of detecting that effect. But you see where all the circles are, which are simulating mutual model landscapes. Most landscapes in the real world and in our simulated world are in a place where it's hard to detect that effect because it isn't limited. So something to think about when doing this kind of research is can you detect the effects. So a seventh challenge, evolutionary population genetics, could be connecting analysis of movement data and genetic data to validate predictions and provide mechanistic explanations. And I'm not going to get into a great deal on this because we saw yesterday how we can use movement data with this conditional logistic regression idea and then verify that the genetic-based resistance surface and one based on movement data are essentially equivalent. So their response to forest cover, middle elevations, or human development. So it's a really useful thing to verify, validate, and compare results using two kinds of data, genetic data and forest. Another grand challenge was to understand how effective population size is different in continuously distributed populations, complex landscapes, then it would be an ideal pandemic populations. So very recently, Dave Sherk wrote a paper using our simulation approach to look at spatially explicit modeling effective population size in continuous populations. We were able to generalize civil rights relationship for neighborhood size, which we developed isolation by distance. We're able to generalize it into an isolation by resistance landscape, so we can use the ideas of effective population size, genetic neighborhoods in landscape genetics context, which could be useful. Another challenge that is very important is a couple simulation and empirical modeling to validate landscape genetic predictions. One paper that did this early on, a few years ago, was Andrew Shirk, using simulation modeling to validate his study of mountains, and found that there's a simulation to control the process and see if our statistical method could identify isolation by resistance as the true process relative to isolation by distance or barriers, very similar to our spurious correlations and inference idea showed earlier, and used the simulation model to show that the method did work. But more recently, Jessica Castillo wrote a paper on pikas and used simulation modeling to verify the result they introduced to their empirical optimization. And this is the empirical model, and this is the model that the simulation was optimized to. And so the empirical optimization based on real data and the result that was produced by the simulation were essentially identical, which showed that they reached a result which was very consistent with the population process that they were doing. So that's a robust way to show that they had achieved a high level of rigor in their model. So one of the most interesting and challenging things in landscape genetics is understanding the interaction of gene flow and selection of complex landscapes. And so recently we modified our landscape genetics simulation model to do more than just gene flow and dispersal. We added selection. And we evaluated these classical models of the time to fixation of a single locus in a two-locus system based on strength of selection coefficients. And found, an ideal equation produces a line like this, and in a parametric simulation, our model reproduces it perfectly, but when we have isolation by distance, there's longer times to fixation, and in some cases, equilibrium conditions that aren't leading to fixation. So, spatial energy, spatial processes fundamentally affect the rates of evolution in the pattern, and this kind of modeling is useful to explore that. And our next step in that process was to look at evolution in a sympatric environment where only resistance to dispersal can lead to the evolution of reproductively compatible genotypes, which can then become incipient species. So the next grand challenge is to understand the effects of climate change and population structure. So what we were able to do in this piece of work was to look at empirical analysis, to understand how landscape resistance for American marten is a function of elevation, which is a surrogate of climate, and then look at climate change effects, and then see how that could affect the connectivity of populations and the genetic structure of populations over time. And so we were able to use the resistant kernel method to map areas that are expected to be connected by dispersal in the current landscape. Essentially, high elevations are connected, and then projecting climate change out to about 100 years from now, applying the resistant kernel to change the resistant surface of that, find a reduction in habitat extent that is connected, and a higher isolation of the random patches, which is kind of fragmentation. Then we use the simulation model, simulation of individual-based gene flow and dispersal, see pop. And we can simulate changes in heterozygosity and nuclear richness over those scenarios, and associate them with changes in the extent and fragmentation of connected habitat. And we find allelic richness is very, very closely related to the extent, almost perfectly correlated with the extent of connected habitat, while heterozygosity has a threshold effect that is related to number of isolated patches. So related more to fragmentation. And we extended that with our tool SGD, which estimates spatially explicit patterns of genetic diversity, and areas in the landscape that have high genetic diversity and how they change across the simulation of future climates to see where and how much reduction in genetic diversity we can expect. And then extrapolate that with some statistical modeling to a very broad area and map expected allelic richness across, this is 30 million hectares. There might be some scaling uncertainties in extending our result of this broadly, but if this relationship is true, we expect very high allelic richness in these high mountain populations here. And then as the climate warms, we see certain areas, like that one, which are expected to have very large reductions in allelic richness because of reductions in the extent of connectivity of habitat. And some areas remain very high in genetic diversity because they remain large blocks connected. So another great challenge, 12, is to connect controlled, replicated experimentation to modeling and empirical optimization. Because experimentation is critical to verify mechanisms. And we want to connect the modeling to extend it across scale. So a recent study we did was on Fremont cottonwood trees, which are riparian trees in the deserts in the southwest of the United States and also in Mexico. And we were modeling how connectivity and gene flow of those trees affected by climate gradients for the networks. So we sampled populations across their American range and modeled gene flow as a function of river network connectivity found in large rivers, promoted gene flow, and small rivers resisted. And we also found that variation in seasonality of precipitation affected gene flow as well. So we combined through a reciprocal causal optimization, the river effect, and the climate effect, find a model that optimizes total explanatory power across all those combinations, and find that the Fremont cottonwood gene flow is controlled by the connectivity of the river network and seasonal precipitation. And you can see the map over here. So now we are implementing what we call Southwest Experimental Garden Array, which is a reciprocal transplant province trial study. It allows us to do multi-factor climate change communications, warming experiments, carbon dioxide oxidification, watering, and drought treatments. And then we can integrate genetics and climate change research. So we can take our understanding of how climate affects gene flow for these cottonwoods, and we can study it and experiment with control environment. And so that is the last of my grand challenges. And as you can see, there are big challenges in this integration. And the biggest one, as I mentioned, is getting people to do experimentation, and people to do modeling toward the depth. And that's just come a long way. For example, Dr. West is a great model, but she's also experimental. She saw an experimental model system example. So there's great power in integrated experimentation, controlled experiments, which allow you to have strong influences, identify causes and mechanisms, and then connect the model, which allows you to extend those implications to broader landscapes, bigger areas, longer periods of time, different contexts. But all of them require data. And in the case of landscape genetics, the new data, genomic data, epigenomic data, integrated with bioinformatics, is a giant area of development and immensely challenging. handle data sets like that. We connect them to experiments. Do experiments that are directly in evaluating variation in genomic and epigenomic characteristics and integrate those with model, extend those across bigger scale ranges with space and time. So that was the end. And I can answer any questions.
¿Alguna pregunta en el auditorio? Thank you very much for your talk. I found it very scary. And there's a question that I have in my head for a long time. When we want to explain some things like the effects of anthropogenic change into landscape or even global warming. And my question is, the information, the genetic information that we can observe with our data, it really can explain those recent events because of the mutation rate and
yeah. So the example I showed you, one of our grand challenges was time lags in landscape change. And it is an important issue because you are right in the fact that there is a time lag. And depending on what method you use, if you use individual-based methods, not using population-based statistics, not just things like correlation between cost distances and genetic distances among individuals. That is much more rapid in changing. But we find still it would take at a short dispersal distance 50 generations to detect that effect. If you're using FST, you can take 200 generations. You won't detect it. With an individual-based method, if you have limited dispersal, this animal or plant does not disperse very much. Take 50 generations. 50 generations is probably longer than the time frame of your studies interest in most organisms. If organisms have very long dispersal abilities, you can detect their effects almost right away. So species that are very, very local and have short lives, you can detect fragmentation effects with genetic data almost instantly with individual-based methods. Now, you can detect fragmentation effects instantly with genetic-based methods if you use different approaches. Looking at parent-offspring detections, you're sampling a large number of individuals, and you can do an analysis to determine their relatedness, determine which ones are parents, offspring. You can then see if they're moving through a landscape across a barrier, across a road, or something like that in real time, because there's no population inertia, because those individuals can be detected, and you know their relatedness. So for immediate observation of recent changes, doing analysis of parent-offspring dispersal is probably the most robust. But you can use these landscape genetic approaches, these individual-based methods, particularly when the organisms are highly mobile and have relatively short generations. You can detect it quickly.
I remember that. Well, thank you very much, Dr.
Bueno, pues, antes de salir, a las personas que están registradas para los talleres, si por favor pueden pasar conmigo nada más para verificar, ¿no? Que sí están, que sí van a ir, que tienen que recibieron el correo con la información de los paquetes que deben instalar en su computadora, porque es importante que ya lo tengan cargado para no perder tiempo el lunes y el martes en ver eso. Entonces, por favor, aquí estoy. Gracias.
Vamos a un receso de 10 minutos y recuerden votar porque después hasta la comida se va a estar la una por sus fotoreportajes favoritos. Gracias.
Hola, ¿qué tal? Hola, bien, ¿tú?